Let's begin by going over exactly what we'll cover in this video. First, we'll explain what monoclonal antibodies are and how they differ from natural antibodies. Next, we'll examine the specific process scientists use to produce these antibodies. Finally, we'll explore their various applications in diagnosis, research, and treatment. Let's start by understanding what monoclonal antibodies actually are. Antibodies are normally produced by our immune system's B lymphocytes, a type of white blood cell. These Y-shaped proteins bind to specific foreign substances, helping our body identify and neutralize threats. When our immune system responds naturally, many different B lymphocytes produce slightly different antibodies. In contrast, monoclonal antibodies are identical copies that all target exactly the same binding site on one specific antigen. This remarkable specificity makes monoclonal antibodies incredibly useful tools. Because they only bind to one particular target, they can be used to identify or attack very specific substances or cells. Now, let's look at how scientists produce these specialized antibodies. The process begins by stimulating mouse lymphocytes to make a particular antibody. This is done by injecting a mouse with the specific antigen the scientists want to target. However, lymphocytes don't survive long in laboratory conditions. This presented a significant challenge when scientists first attempted to produce antibodies artificially. The breakthrough came when researchers discovered they could fuse these antibody producing lymphocytes with tumor cells to create what's called a hybridoma cell. This hybrid cell inherits two crucial characteristics: the ability to produce a specific antibody from the lymphocyte and the ability to multiply rapidly and indefinitely from the tumor cell. After creating these hybridoma cells, scientists isolate individual cells and allow them to multiply, creating clones. Each clone produces identical antibodies, all targeting the same antigen. The resulting population of identical cells all produce the same antibody. Scientists can collect these antibodies and purify them for various applications. Let's now explore some of the important uses of monoclonal antibodies. One common diagnostic application is pregnancy tests. These tests contain monoclonal antibodies that specifically bind to a hormone called HCG, which is only present in the urine of pregnant women. When a woman uses a pregnancy test, her urine moves along the test strip. If she's pregnant, the HCG in her urine binds to the antibodies, creating a visible line or symbol on the test. In medical laboratories, monoclonal antibodies are used to measure levels of hormones and other chemicals in blood samples. They can also detect specific pathogens, making them valuable tools for disease diagnosis. Research scientists use monoclonal antibodies to locate specific molecules within cells or tissues. By attaching a fluorescent dye to the antibodies, researchers can see exactly where their molecules of interest are located in a sample when viewed under a microscope. Perhaps one of the most significant applications is in treating disease, particularly cancer. Cancer cells often have specific markers on their surface that aren't found on normal cells. Monoclonal antibodies can be designed to recognize these unique markers. These therapeutic antibodies can work in several ways. They might be bound to a radioactive substance, allowing targeted delivery of radiation to cancer cells. Alternatively, they could carry a toxic drug that kills the cancer cells once the antibody attaches to them. Some monoclonal antibodies are linked to chemicals that stop cells from growing and dividing. In each case, the antibody delivers its payload specifically to cancer cells, minimizing damage to healthy tissue. This targeted approach differs significantly from traditional cancer treatments like standard chemotherapy and radiotherapy, which affect many body cells in addition to the cancer cells. Monoclonal antibodies precisely target specific cells, potentially reducing side effects. Despite their usefulness, monoclonal antibodies do have limitations. They can cause side effects that weren't initially expected. When first developed, scientists thought that because they target specific cells so precisely, they would have very few side effects. Additionally, the development of new monoclonal antibody treatments is costly and time consuming. This means they aren't as widely used in treatment as scientists originally hoped they would be. Understanding monoclonal antibodies helps us appreciate how scientific innovations can harness natural biological processes to create powerful tools for medicine and research. These laboratory created molecules continue to play an increasingly important role in diagnosing and treating a range of conditions. In our next episode, we'll explore how plants respond to pathogens and the various defense mechanisms they've evolved to protect themselves from disease.